Today we're taking a fascinating journey into a part of your brain that, well, quietly orchestrates so much of your daily life, the reticular formation in the brainstem. Now, it might sound like some ancient automatic system just kind of humming in the background.
Yeah, people often overlook it.
Right, but trust me, this network is surprisingly central to a vast range of functions, way beyond just, your basic reflexes.
Absolutely. We tend to think of the wrinkly outer layers, the cortex for complex tasks, but this older, seemingly simpler structure, the reticular formation, it's absolutely vital.
So what sources are we looking at?
For this deep dive, we've got some really detailed sections from a neuroanatomy textbook. It breaks down how the reticular formation is organized, the surprisingly diverse roles it plays, and its extensive connections throughout the nervous system.
Okay, so our mission is to really unpack these core functions and reveal why this network is so incredibly essential. We're talking about everything from how you drift off to sleep at night to how you perceive the world around you, and even those gut feelings.
The reticular formation plays a role.
Which is pretty wild. So if you're looking to get a solid understanding quickly and maybe have a few of those aha moments, you're definitely in the right place.
Should be interesting.
Okay, let's get into the details. Organization first.
Yeah, let's start there. So the name reticular formation itself gives you a bit of a visual from its early days under the microscope.
Reticular meaning net-like.
Exactly. It refers to this polysynaptic network, meaning it involves multiple connections between neurons, not just direct point-to-point links located right there in the brainstem.
Okay.
But it's crucial to understand its reach. It extends upwards into the thalamus and hypothalamus, and also downwards into the spinal cord's own intricate network.
So it's not just contained in the brainstem.
No, not at all. And while reticular came from that early net-like appearance, over time, with better tools, we've been able to identify more and more specific clusters of neurons, or nuclei, within this network. It's more structured than the name might suggest.
Right, so it's not just one big, indistinct area. Our sources highlight three main subdivisions, almost like distinct neighborhoods with their own specializations. First, there's the median reticular formation right in the center. This is home to the Yaphe nuclei.
Yes.
Which are a major source of serotonin projections that spread throughout the entire central nervous system.
And what's truly remarkable is just how far these serotonergic neurons reach. The textbook points out they have the most widespread distribution of any group of neurons in the CNS.
Wow. Everywhere.
Pretty much. They send projections all the way up into the cerebral hemispheres, throughout the brainstem itself, into the cerebellum, and even down into the spinal cord. Essentially, serotonin-releasing axons are found, permeating all the gray matter.
Okay, and connecting that to everyday experience.
Well, this vast serotonin network helps explain why things like, say, a lack of sunlight or changes in our diet can have such a profound impact on our mood. It's affecting this fundamental neuromodulatory system that touches almost every part of our brain.
Makes sense. Okay, so that's the median part. What's next?
Then right next to it, we have the paramedian reticular formation. This area is characterized by its larger neurons, called magnocellular neurons.
Magnocellular, big cells.
Right. And in the lower pons and upper medulla, you even find some really big ones, gigantocellular neurons. Our source tells us this is primarily an efferent system.
Efferent meaning sending signals out.
Exactly. It's mainly sending signals out, both upwards towards the midbrain and thalamus, and downwards towards the spinal cord. Interestingly, though, it also receives a significant amount of input from the cortex itself through corticoreticular fibers.
Ah, so there's a feedback loop, or at least communication, with the higher brain areas.
Precisely. This connection back to the cortex emphasizes that the reticular formation isn't just operating in isolation, it's in constant communication with and being influenced by those higher brain regions.
Okay, and the third part.
Finally, the outermost layer is the lateral reticular formation. This is parvocellular.
Smaller cells this time.
Yep. smaller cells. And this subdivision is primarily afferent. It's the main receiver of sensory information arriving at the reticular formation.
And when they say all sensory pathways, they really mean it, don't they?
They do. Our textbook lists olfactory input, that's smell, coming in via the medial forebrain bundle, visual information from the superior colliculus, auditory input from the superior olivary nucleus, vestibular signals related to balance from the medial vestibular nucleus,
Right.
and all sorts of somatic sensory information, touch, pain, temperature coming in from the spino-reticular tracts and trigeminal nuclei.
Wow. So think of the lateral reticular formation as like the brainstem central information hub.
That's a great way to put it. All these pathways are like different phone lines, bringing in updates from all our senses: sight, sound, touch, balance, even smell has a direct line.
And what happens to that information once it arrives?
Well, what's important to appreciate is that while the lateral part is primarily receiving, its axons then extensively branch out and connect with those larger magnocellular neurons in the paramedian reticular formation we just talked about.
Okay, so it sets up a relay.
Exactly. A crucial relay point where sensory input can influence motor and other output functions mediated by that paramedian region. The text also mentions that some of these lateral axons act as pattern generators for cranial nerve nuclei, contributing to things like, facial movements.
Pattern generators. We'll come back to those, I think.
We will. But first, woven throughout this whole intricate structure are some incredibly important chemical messengers: the aminergic neurons.
Aminergic, using adenine neurotransmitters.
Correct. Neurons that use neurotransmitters synthesized from aromatic amino acids. We're talking about serotonin again, of course, but also dopamine, norepinephrine, epinephrine, and histamine. These tiny chemical signals have enormous effects across the brain.
Okay, let's break those down. Serotonin we touched on.
Right. We've discussed its widespread reach, and as the source emphasizes, its dysregulation is clinically linked to major depression, a huge area of research.
And dopamine.
Then we have dopamine, with key clusters of neurons in the substantia nigra famously involved in movement control. Think Parkinson's disease.
All right.
And also in the ventral tegmental nuclei, or VTA, which project to the frontal lobe and the nucleus accumbens brain areas, critical for reward, motivation, addiction.
The mesolimbic pathway.
Exactly, the mesocortical and mesolimbic projections. Crucial stuff.
And norepinephrine. That's another big player, isn't it?
A very big player.
Yeah.
Our source indicates that about 90% of its cell bodies are located in the locus ceruleus, a small area in the upper pons.
Just one small spot.
Primarily, yes. But these neurons send their projections in pretty much every direction. Up, down, sideways.
So it's like a broadcast system.
Indeed. Think of norepinephrine as a global modulator, influencing things like attention, alertness, vigilance, and even the body's response to stress.
Okay. What about epinephrine? Adrenaline.
Epinephrine-secreting neurons, on the other hand, are less numerous. They're primarily found in the medulla, projecting mainly to the hypothalamus and the spinal cord.
Less widespread than norepinephrine or serotonin?
Definitely. And finally, histamine neurons, located in the tuberomammillary nucleus of the hypothalamus, actually also play a significant role in promoting arousal and wakefulness.
Interesting. Now, what's really intriguing is that the effects of all this aminergic activity, say in the cortex, aren't always straightforward. It's not just like flipping a switch, is it?
No, not at all. Our textbook points out that for each of these amines, serotonin, dopamine, norepinephrine, there's often more than one type of receptor on the receiving neurons, sometimes many types.
So the same chemical can have different effects depending on which receptor it hits.
Precisely. Plus, some of these aminergic neurons also release peptides alongside the main neurotransmitter. These peptides can kind of fine-tune or prolong the action of the primary amine.
Adding another layer of complexity?
Absolutely. It's a crucial point. The brain is an incredibly complex system, and these aminergic systems act like conductors with a vast orchestra of instruments, using subtle cues to influence the overall sound,
I like that analogy.
And when you consider that a single cortical neuron can receive thousands of other inputs, you start to see how the effect of activating just one type of aminergic receptor can vary depending on the neuron's overall state at that moment.
So, while we know these aminergic systems are essential for many behaviors, mood, arousal, attention, the precise mechanisms are still, well, they're still being worked out.
Exactly. It's a very active area of research.
Okay, so we've explored the organization of this network, the different regions, the chemical messengers. Now let's dive into what it actually does, the functional anatomy.
Right. What are the jobs?
Our source has a really helpful table outlining the diverse range of these functions. Let's maybe start with pattern generators.
Good place to start. This is where the reticular formation demonstrates its involvement in those automatic, rhythmic activities we often take for granted.
Right.
Think about conjugate eye movements, where both of your eyes move together smoothly in the same direction. There are premotor nodal points within the midbrain and pons that coordinate this. And they're connected to the cranial nerve nuclei that actually control the eye muscles. It ensures your eyes track together.
Okay, so coordinating eye movements. What else?
And it's not just eye movements. The source mentions rhythmic chewing. There's a specific nucleus, the supratrigeminal premotor nucleus, located in the pons, that handles that pattern.
Chewing, another automatic rhythm.
Exactly. And then further down the medulla, there are separate premotor areas responsible for essential reflexes, like swallowing, vomiting, coughing, yawning, and sneezing.
Wow, all those basic bodily functions.
All those things your body just does without you consciously thinking about them. They're generated or coordinated here.
Our source even mentions separate sections on locomotor pattern generators.
Right. Those are primarily in the spinal cord, but influenced from the brainstem.
And higher level bladder controls also influenced by the reticular formation?
Correct. The pons has key centers for bladder control.
Right.
Oh, and let's not forget the salivatory nuclei in the pons and medulla.
For saliva.
Saliva, yes.
Yeah.
And even tears through parasympathetic nerve fibers. So quite a range of automatic patterns.
Okay, moving on to another absolutely critical function, respiratory control. Breathing.
Yes, fundamental. The medulla, the lowest part of the brainstem, houses the dorsal and ventral respiratory nuclei.
Dorsal and ventral. Back and front.
Sort of, yeah. The dorsal nucleus is primarily responsible for inspiration breathing in. It sends signals to the muscles like the diaphragm and intercostals that expand our chest. And crucially, it receives vital input from chemoreceptors that constantly monitor the levels of gases like CO2 and oxygen in our blood and cerebrospinal fluid.
So it knows when we need to breathe, more or less.
Exactly. It adjusts based on those chemical signals.
Yeah.
Now, the ventral respiratory nucleus is mainly involved in expiration, breathing out.
Okay.
What's interesting is that it's relatively inactive during quiet, normal breathing. Expiration is mostly passive then. But it really kicks in during more forceful breathing, like when you're exercising or singing loudly or coughing.
Ah, so for active exhalation.
Precisely. Our source also highlights the medial parabrachial nucleus up in the pons. This seems to play a role in our breathing patterns, specifically when we're awake.
Okay.
And it has a fascinating link to hyperventilation experienced during anxiety or panic attacks through its connections with the amygdala, the brain's emotional center.
Wow, so anxiety can directly impact breathing via this pathway.
It seems so. The connection is there.
And those chemoreceptors you mentioned, how do they work?
Well, the system is remarkably sophisticated. There's the medullary chemosensitive area right there on the surface of the medulla. It's highly sensitive to the concentration of hydrogen ions in the cerebrospinal fluid.
Hydrogen ions reflecting CO2 levels.
Exactly. It directly reflects the carbon dioxide levels in our blood. If CO2 levels rise, meaning acidity increases, it stimulates that dorsal respiratory nucleus to increase our breathing rate and depth to expel more CO2.
Makes sense. Get rid of the excess CO2.
Right. Then you also have the peripheral chemoreceptors, primarily the carotid body's tiny sensors located near the carotid artery in your neck.
Okay.
These respond directly to low levels of oxygen or high levels of carbon dioxide in the arterial blood itself. When they detect changes, they trigger reflexive adjustments in our breathing rate via the brainstem.
So multiple systems monitoring blood gases.
Yes, ensuring our breathing matches the body's metabolic needs. The source notes that the aortic chemoreceptors, located near the aorta, seem less significant for this in humans compared to the carotid bodies.
Got it. Okay, another absolutely vital autonomic function handled nearby is cardiovascular control, regulating our heart rate and blood pressure.
Also critical. Our textbook explains the role of the baroreceptor center, which is located in the solitary nucleus, again in the medulla.
Solitary nucleus, again, it seems involved in a lot.
It is. It's a major integration center for visceral sensory information. In this case, it receives input from specialized stretch receptors called baroreceptors found in the walls of the carotid sinus and the aortic arch.
Baro meaning pressure, so pressure receptor.
Exactly. These receptors constantly sense the stretch in those major arteries, which tells the brainstem about your current blood pressure.
And what does the brainstem do with that info?
What's truly ingenious are the baroreceptor reflexes. When your blood pressure rises, say, these reflexes immediately trigger both parasympathetic and sympathetic responses to bring it back down to a normal range.
How does that work?
The parasympathetic response, via the vagus nerve, acts to slow down your heart rate. At the same time, the sympathetic response is inhibited, which causes a reduction in the constriction of your peripheral arteries, lowering resistance and allowing blood to flow more easily.
So it adjusts heart rate and blood vessel tightness.
Right. It's this continuous balancing act that helps maintain stable blood pressure, ensuring adequate blood flow to the brain and other vital organs.
Like when you stand up quickly.
Perfect example. When you stand up quickly, gravity pulls blood downwards, your blood pressure might momentarily dip. That's when these baroreceptors sense the reduced stretch, like tiny pressure sensors. And they signal the brainstem to kick in the sympathetic system, get the heart pumping a bit faster, tighten the vessels slightly to compensate and keep you from feeling faint. It's all happening automatically, constantly, thanks to this medullary system.
Incredible. Okay, let's shift focus now to something we all experience every single day, hopefully. Sleeping and wakefulness.
Ah, yes, the sleep-wake cycle. A huge role for the brainstem and interconnected structures here.
Our source delves into the different EEG patterns that characterize these states, the electroencephalogram readings.
Right. You see these fast, irregular, low-amplitude waves when we're awake and alert. Then, as we fall asleep, the waves get slower, more rhythmic, and higher in amplitude.
That's non-REM or slow wave sleep.
Exactly. Stages of non-REM sleep. And then, periodically through the night, we enter REM sleep, rapid eye movement sleep.
Where the EEG looks more like wakefulness again.
Yes, it's more desynchronized, similar to being awake, but you have muscle atonia, rapid eye movements, and often vivid dreaming.
And we cycle through these stages.
Yes, the cyclical nature of these different sleep stages is really key. And the textbook introduces this helpful concept of flip-flop mechanisms to explain the transitions.
Flip-flop, like a switch.
Kind of. There's essentially a switch, a mutually inhibitory circuit, that flips between being awake and being asleep. And there's another similar switch that flips between non-REM and REM sleep. This allows for those relatively rapid transitions between these quite distinct brain states.
What drives the main sleep-wake flip-flop?
Well, normal sleep is driven by a combination of factors. There's our homeostatic sleep drive, basically. The longer you're awake, the more sleep pressure builds up.
Makes sense.
Then there are our circadian rhythms governed by the suprachiasmatic nucleus, the SCN, in the hypothalamus. That operates on a roughly 24-hour cycle, influenced by light.
Our internal clock.
Right. And finally, there are allostatic factors, which relate to things like our nutritional state, stress levels, illness, things that can override the other two drives temporarily.
Okay, so what parts of the brainstem are actually involved in keeping us awake? The arousal system.
The system that promotes wakefulness, often called the ascending arousal system or AAS, is primarily located in the caudal, midbrain, and rostral pons, so upper brainstem. Our source highlights cholinergic neurons using acetylcholine from areas called the pedunculopontine nucleus, PPT, and the laterodorsal tegmental nucleus, LDT.
Okay, cholinergic neurons, what do they do?
A key action is inhibiting GABAergic neurons in the thalamus. The thalamus is like a gate for sensory information heading to the cortex. By inhibiting these inhibitory thalamic neurons, the PPT-LDT essentially opens the gate, clearing the path for sensory information to reach the cortex and keep us alert.
Disinhibition. Makes sense.
And then you have that whole network of monoaminergic neurons playing a crucial role in arousal as well.
Right.
We talked about them earlier.
Right. Norepinephrine, serotonin.
Norepinephrine from the locus ceruleus, serotonin from the raphe nuclei, glutamate from the parabrachial nucleus, dopamine from the periaqueductal gray matter, PEG, and histamine from the tuberomammillary nucleus in the hypothalamus.
All contributing to wakefulness?
Yes, they all send their projections widely to the basal forebrain and the cortex, contributing to our overall state of alertness and responsiveness. The text also mentions other contributing players, like orexin or hypocretin neurons in the hypothalamus, which strongly promote wakefulness, and certain cholinergic and GABAergic neurons in the basal forebrain itself. It's a distributed network.
Okay, so that's the wake side of the switch. What about the sleep side?
On the other side of the coin, we have the system that actively promotes sleep, located primarily in the hypothalamus, particularly the preoptic area.
Hypothalamus, again, seems key for regulating basic drives.
It really is. The ventrolateral preoptic nucleus, or VLPO, uses inhibitory neurotransmitters like GABA and galanin to actively inhibit those wakefulness-promoting centers we just discussed. The locus ceruleus, raphe, tuberomammillary nucleus, PPT-LDT.
So it actively shuts down the arousal system.
Exactly. And the nearby median preoptic nucleus, MNPO, seems to be responsive to our body's homeostatic need for sleep, that accumulating sleep pressure. It can then activate the VLPO neurons when it's time to rest.
And are there sleep-promoting centers in the brainstem, too?
Yes. The source also mentions GABAergic neurons in the caudolateral pontine reticular formation that appear to have sleep-promoting effects, possibly by inhibiting arousal regions, too.
So this flip-flop switch analogy really helps visualize this interplay, doesn't it?
It does. During sleep, the sleep-inducing system, mainly VLPO, actively inhibits the arousal system, keeping us in a state of slumber. Conversely, when we're awake, the arousal system inhibits the sleep-inducing circuits, VLPO, preventing us from drifting off.
Mutual inhibition.
Exactly. It's a reciprocal inhibition that allows for relatively stable states. You're either mostly awake or mostly asleep, and also allows for those quick transitions when needed, like waking up suddenly to an alarm.
Orexin neurons also help stabilize the awake state.
And there's a similar flip-flop for REM and non-REM sleep.
Yes, a similar flip-flop switch exists between the REM-on and REM-off centers located primarily in the pons. REM-on cells, cholinergic and glutamatergic, promote REM sleep features, while REM-off cells, aminergic and GABAergic, promote non-REM sleep. They mutually inhibit each other, allowing for the shifts between these sleep stages throughout the night.
And disruptions here can cause problems. Absolutely. The source briefly touches on how disruptions in these delicate systems, lesions, chemical imbalances, can lead to various sleep disorders, like insomnia, narcolepsy, or REM sleep behavior disorder, highlighting the critical importance of this brainstem and hypothalamic regulation for healthy sleep.
It's maybe worth clarifying something here. We hear about the reticular activating system, or ARIs sometimes. Is that the same as this arousal system?
That's a good point. The older term was indeed the ascending reticular activating system, or ARIS. But our current understanding is that the ascending arousal system, the ARIS, isn't solely the reticular formation anymore.
Okay.
It involves a broader network of interconnected brain regions. Yes, key reticular formation nuclei like the locus ceruleus, raphe nuclei, and the PPT-LDT are core components. But it also critically involves the tuberomammillary nucleus in the hypothalamus, contributions from the basal forebrain, thalamus, and even cortex. So the reticular formation is a critical component, but the AAS is a larger, more distributed system than just the RF itself.
Good clarification. Okay, finally, let's delve into sensory modulation and specifically this idea of gate control theory.
Right. Gate control. This theory, originally proposed by Melzack and Wall, describes how the transmission of sensory information, particularly pain signals, can be regulated or gated, turned up or down at different levels of the nervous system.
Like a volume control for sensation.
Sort of, yeah. The source mentions tactile sensory gating happening at the dorsal column nuclei, where touch information first relays in the brainstem. But the primary focus here, and where the theory is most developed, is on nociceptive transmission, the signals that tell us about potential or actual tissue damage, what we perceive as pain.
Okay, so gating pain signals, where does this happen?
A key location is in the dorsal horn of the spinal cord's gray matter, and a similar area called the spinal trigeminal nucleus for pain signals coming from the head and face. Within these areas, there's a specific region called substantia gelatinosa.
Substantia gelatinosa, okay.
This area is packed with networks of interneurons, small neurons that connect other neurons locally. Some are excitatory, using glutamate, and others are inhibitory, using neurotransmitters like GABA and enkephalin, an opioid peptide.
And the pain signals coming in?
We have different types of nerve fibers that carry pain signals from the body. There are the faster, thinly myelinated A delta fibers responsible for that initial, sharp, well-localized pain.
Like a pinprick.
Exactly. And then there are the slower, unmyelinated C fibers that transmit that longer-lasting, dull, burning, poorly localized ache or pain. These C fibers often release a neurotransmitter called substance P, in addition to glutamate, which tends to amplify and prolong the pain signal in the dorsal horn.
Okay, so different fibers for different pain qualities. How does the gate work at this level?
Well, one key aspect is called segmental antinociception, anti-pain mechanisms operating at the same spinal cord level where the pain signal enters. The theory proposes that when large A-beta mechanoreceptor fibers, the ones that respond to normal light touch and pressure, not pain, are activated, they can stimulate those inhibitory interneurons, the ones using GABA and enkephalin, in the substantia gelatinosa.
The touch fibers activate the inhibitory cells.
Right. And these inhibitory cells then act to suppress the activity of the spinothalamic relay cells, the neurons that are responsible for carrying the pain signals up the spinal cord towards the brain. This inhibition can happen both presynaptically, reducing neurotransmitter release from the pain fibers, and postsynaptically, making the relay cell less likely to fire.
So activating touch fibers closes the gate to pain signals.
Essentially, yes. This is the fundamental reason why rubbing a sore spot or bumping your elbow and immediately rubbing it can provide some temporary pain relief. You're activating those A-beta fibers.
And 10S units.
It's also the principle behind the use of 10S units transcutaneous electrical nerve stimulation. They use electrical currents to selectively activate those large A-beta fibers, aiming to close the pain gate.
Fascinating. Okay, that's segmental control. What about control from higher up, supraspinal?
Right. Then we have supraspinal antinociception, which means that higher brain centers can also exert control, sending signals down to influence how we perceive pain at the spinal cord level.
Descending modulation.
Exactly. The Magnus-Raffin nucleus, MRN, one of those Raffin nuclei we mentioned earlier, located in the medulla, plays a significant role here.
The serotonin source.
Yes. Its serotonergic projections descend down the spinal cord to the substantia gelatinosa. There, they can excite those inhibitory enkephalin-containing interneurons, effectively reducing the transmission of pain signals upwards and leading to analgesia, or pain relief.
So stimulating the MRN can reduce pain.
Correct. Studies show that stimulating the MRN can produce profound analgesia. Also, norepinephrine projections originating from areas like the locus ceruleus in the pons and other nuclei in the medulla also descend and appear to have a direct inhibitory effect on those spinothalamic pain relay neurons.
So both serotonin and norepinephrine pathways coming down can dampen pain.
Yes, they form a powerful descending pain control system.
What's this about diffuse noxious inhibitory controls? Sounds complicated.
It's a particularly interesting concept. The magnus raphe nucleus and other brainstem centers involved in pain modulation actually receive input from spinal-reticular neurons, neurons carrying signals up from the spinal cord that are responding to noxious or painful stimuli happening elsewhere in the body.
So pain in one area can trigger descending inhibition.
That seems to be the idea. It helps explain how applying a painful or intensely uncomfortable stimulus in one area, like strong pressure or acupuncture needles, can sometimes relieve pain in a completely different, unrelated area. It's sometimes called counter-irritation. The noxious input activates the descending pathways, which then have a more widespread inhibitory effect.
Well, maybe part of how acupuncture works.
It's thought to be at least one potential mechanism, yes. Furthermore, the magnus raphe nucleus is itself strongly influenced by the periaqueductal gray matter, or PAG, in the midbrain.
PAG involved in dopamine earlier, now pain.
Yes, it's a key node. Stimulated the PAG can activate the MRN and produce powerful analgesia. This PAG-RIF connection has been explored for pain management therapies, like deep brain stimulation in some intractable pain cases.
And finally, the source touches on stress-induced analgesia, like not feeling pain in an emergency.
Right. This seems to involve the PAG as well. Normally, the PAG's ability to activate the descending pain control pathway via the MRN is kept in check, kind of tonically inhibited, by local GABAergic interneurons within the PAG itself.
So GABA neurons are putting the brakes in the pain relief system.
Exactly. However, opioid peptides, like beta-endorphin released during stress, can inhibit these GABAergic brake neurons.
Inhibiting the inhibitors.
Yes, which essentially releases the brakes on the PAG output neurons, allowing them to fire more strongly, activate the MRN, and suppress pain transmission. This could be why people sometimes don't feel injuries immediately in dangerous or highly stressful situations.
Wow, that's a complex cascade.
It is. And the amygdala, our brain's fear and emotion center, can also directly stimulate the PAG in fearful situations, contributing to this stress-induced pain dampening effect.
So multiple ways the brain can shut down pain signals when needed.
Definitely. The source also briefly mentions that there's further gating of sensory information happening up at the thalamic level, and that the anterior cingulate cortex, part of the frontal lobe, plays a critical role in processing the emotional, unpleasant, aversive quality of pain, rather than just its location or intensity.
So pain perception is multi-layered.
Very much so. And just as a quick aside, the source includes a clinical note about urge incontinence, highlighting the involvement of bladder control centers, particularly in the pons, like Barrington's nucleus, and noting how monoamines like serotonin and norepinephrine influence their function, which ties back to some therapies used for bladder issues.
Okay, covering a lot of ground there.
It really does. So as we kind of wrap up this deep dive, it becomes really clear, I think, that the reticular formation is so much more than just some primitive automatic network humming away in the back of our brain.
Yeah, definitely not just reflexes.
It's incredibly intricate organization, these different nuclei, the widespread connections, the various neurotransmitters. It all allows it to play absolutely crucial roles in a vast spectrum of functions.
Right, from those basic reflexes we rarely think about, like breathing and swallowing,
To fine-tuning our sensory experiences, like that pain gating,
governing our states of consciousness, sleep, and wakefulness.
and even influencing our behavioral responses to the world around us through connections with emotion and motivation centers is deeply integrated.
Absolutely. This seemingly old part of the brain underpins so many of our fundamental daily experiences in ways we might never consciously appreciate. It really makes you think, doesn't it? About how even subtle disruptions within these fundamental networks may be due to injury, disease, or even chemical imbalances could potentially manifest in such a wide range of neurological and psychiatric conditions. Everything from sleep disorders to chronic pain, attentional problems, mood disorders.
It underscores the delicate balance.
Exactly. It highlights the delicate balance and vital importance of this often overlooked but clearly essential brain region. Definitely give you something to ponder. Thanks for joining us on this deep dive.